was successful. So there are key terms that you need to recall. Amphiphilic, that is one having one surface consisting of a hydrophilic amino acids, and the opposite surface, which consists of hydrophobic or water heating or lipophilic ones. Then you have hydrophilic,
both
having an affinity and able to absorb or be wetted by water, and this is water loving. So the outside side of the cell, which is exposed to the extracellular fluid, is called hydrophilic, and the inside part of the
na left relax nini bana
or be form a
Maserati.
yes
Hajafika? Hajafika huyo. Sema
nitamwambia. Yes.
Aliona viatu. Aliona viatu yangu hapo na bahati sana. Walikuja kitu kweli? Walikuja kitu?
Nimekuja exam.
Exam ilikuwa rahisi. Nilikuwa naambia Devi tense hapa na anatuma notes kama karibu 1000. Bado iko? Bado iko.
It's called Singer and Nicholson. So it's a model of the structure of the cytoplasmic membrane that describes the membrane as a lipid bilayer, not of proteins which are distributed in a mosaic-like pattern on the surface of the cells in the membrane.
Pano umefanyaje?
The fluid model describes the plasma membrane structure as a mosaic of phospholipids, cholesterol, proteins, and carbohydrates. I think you recall that you have cholesterol, which is inserted in the plasma membrane so that it can assist in very cold and very hot temperatures to regulate the integrity of the plasma membrane. So basically, you have the very
Hello, Prof, we can't hear you.
Mucha missing.
Naumia sana. Hizi panguza maziwa hivi bwana. Hizi na hapo binesha.
Kitu itabidi yosha.
Hapa. Kumwagia maji hakuna namna.
Hata hiyo ni pia ndani. Sikia huu Lex sasa.
Ndio ndio bundles bana, acha mchezo.
Acha niweke class. Mwambie Mike ampigia mwambie atumie bundles.
Atumie bundles.
Anunue bundles.
Carbohydrates attached, and these are the glycoproteins. And then, of course, you have cholesterol, which is inserted in the plasma membrane. It's not necessarily under the glycoproteins. You can find it in here or even in here. So this is the diagram. We also learned that lipid molecules contains a phosphate head and hydrophilic, and then L, which is fatty acid, and it is non-polar and hydrophobic, hence repelling.
Hii ni wifi, si ati acha kutudanganya hapo. Ati anakuwa logged out. Anakuwa logged out. Sijui ni juu ya connection.
Lecturer. Then host. Then host, sindio?
Na anakuwa logged out.
You are now unmuted.
Hiyo ni kutumia bundles ya shule wa.
So, membrane fluid is of protein with restrictions the so the channel proteins, the molecule, each of them are arranged in an amphipathic structure, and that means that the ionic and highly polar groups protrude from the membrane into the aqueous phase. You already know the aqueous phase is either extracellular or intracellular, and the non-polar groups largely buried in the phobic interior of the membrane. So these globular molecules are partially embedded in the matrix of the phospholipid. The bulk of them, of the phospholipid, is organized as a discontinuous fluid bilayer, although small fractions of it, of the lipid, may interact specifically with membrane proteins. And I showed you that with the first slide where I showed you insertion and all the little amino acids, and some are on the outside and some are on the inside. The fluid structure is therefore analogous to a two-dimensional oriented solution of integral proteins or lipoproteins, whatever the case may be, in the viscous phospholipid bilayer solvent. So the lipid bilayer is amphipathic, having hydrophobic and hydrophilic regions of dual solubility the hydrophobic regions, which are concentrated in the membrane interior, and these create a barrier that prevents free movement of polar molecules across the membrane. You then also have the hydrophilic regions, and these face the watery polar surface of the membrane inside. So the amphipathic, you have the polar side, and then you have these are saponifiable. In other words, you can break them up with soap detergents, and these contain fatty acid. And it is this principle, because remember I was trying to explain to you in the last lecture that these the lipid bilayer is the exact thing that covers the coronavirus. And one of the things that you hear everyone is to make sure that you wash your hands with soap. So this is what you're breaking up. I just thought you might want to know that little detail. The non-polar chains, of course, have the carbon atoms, and these are non-saponifiable. They do not contain fatty acid. And then, of course, you have steroids. Sorry, the image is not showing up. It is the 4 carbon chain of cholesterol, which is also included in the So the lipid classification and structure, you have membrane lipids, and these membrane lipids contain phospholipids or glycolipids. The phospholipids contain phosphoglycerides and sphingolipids, and I think through this earlier in the very first lecture that was successful. In this category, you have the fatty acid chain, the phosphate, and the alcohols. Remember these, the ethanolamine, for example. And then sphingosine is found in sphingolipids with the fatty acid chain, the phosphate, and then this is choline. And you also have sphingosine with the fatty acid, which have glucose or galactose. So there are two types, the phospholipid types and the glycolipids type, the glycolipid types. For some reason that's not coming. So the lipids which are found in membrane are 4 types. They're called the phosphoglycerides, the sphingolipids or glycolipids, the glycosphingolipids, and the 4th type is known as the sterols. And all of this is included in a chapter which I've given you to read in your reference materials for this unit. I'm not going to go into details, except tell you that the phosphoglycerides are the most abundant in lipid membranes. They are composed of 2 fatty acid chains, which I've shown you in structure, a phosphate group, an alcohol or glycerol group, and they are amphipathic. That means they are polar and non-polar in the structural conformation. The sphingolipids or glycolipids consist of a lipid with attached carbohydrate groups. The backbone of the structure is called sphingosine. This is a serine which is attached to fatty acid. It has an amino group of the serine which attaches to a second fatty acid chain of the hydroxy group of serine. If the hydroxy group is unbound, it is called a ceramide. if the hydroxy group can attach to a phosphate alcohol molecule, it is called sphingomyelin. And this is the type that covers the nerve cells so that you don't get cascading of a nerve impulse causing electric shock as it travels from one point of the body to another, causing electric shock to all the organs on the way. And sphingomyelin has two fatty acid chains, a serine, a phosphate group, and a choline. I think you're beginning to see the pattern here. There's nothing very complicated about it. Then hydroxy group can also attach to a carbohydrate group, which is then called a glycosphingolipid. So lipids in membranes also include glycosphingolipids, which have two functions. The first is in membrane stabilization, and also they function in nerve cells in the gangliosides, which you did in anatomy, I believe, and also physiology. There are human diseases which are related to malfunction in the metabolism of glycerol, such as neurological disorders such as Tay-Sachs disease. You know, those people who blink uncontrollably, and there are several other physiological ways to tell when someone has Tay-Sachs disease. And sterol is based on the framework of the 4-carbon ring of cholesterol. Molecules disturb the close packing of the hydrocarbon chains of membrane phospholipids. And also membranes containing cholesterol remain fluid at low temperatures. They don't jam together and crack because of the low temperature, and they don't freeze into non-fluid form. So you don't have your cells being converted to ice, experiencing very low temperatures. And then you have cholesterol, which is the stability and mechanical stability of the membranes. And you can to stabilize the lipid bilayer. So phospholipids is the major structural components. I gave you the structure, and you should know that it has two tails, and this is the double bond in here. And in here, the key component is the phosphate. Then you have the choline, the ethanolamine, the serine, the inositol, the glycerol, the glycerol. Phosphatidylamine, P, the ethanol, and so all these. You're reading assignment. Somebody else is reading me with conversation. So you have the lipid anchor proteins. So here is the protein that anchors with the lipid bilayer. And you have a peromembrane proteins outside on the extracellular side. You also have a peromembrane protein on the inside. The integral membrane proteins, as we've learned, cross the lipid bilayer way through. Then you have this hydrophospholipid there and there. And this is the hydrophilic fatty acid side chain, and those constitute the phospholipid bilayer. I know I'm tending to sound repetitive, but you will find out why as we go on. Useful plasma membrane has very key functions. The plasma membrane. Remember, this is and membrane, and that surrounds the cell. We're not talking about mitochondrial membrane. We're not talking about somal membrane. We're not talking about nuclear membrane. We're not talking the entire cell. And the function protein activity, also very important. In general, you have the phospholipid bilayer. All membranes are phospholipid bilayers with embedded proteins. This now includes all those others that I mentioned: the ribosomal membrane, Golgi apparatus membrane, the nuclear membrane, the vesicular membrane, the membrane covering the mitochondria. All those organelles are covered in on the outside, and the main purpose is for the shape and to control movement in and out of those organelles, as well as the cell in general. Now it's not moving, slides are not moving. So embedded in the bilayer proteins, most of the membrane's functions are accomplished by these embedded proteins. We know now there's the integral membrane proteins that go right through, and also the peripheral proteins that are on either side, either in the intracellular side or the extracellular side of the membrane. We know about the glycoproteins and the glycolipids. These proteins and lipids which have short chain carbohydrates, note the short, they're not really long carbohydrates attached on the extracellular side of the membrane. So you will not likely find glycoproteins on having the carbohydrate on the intracellular side. It will more likely be on the extracellular face of that integral membrane protein. So here is an image. I think I've shown you this before. So you can see the carbohydrates here. Side, and then of course all these components on the extracellular domain. Hello, can you hear me? All these components on the extracellular domain interact with other extracellular cell components, and then on the intracellular side you find them interacting. The integrin microtubules or microfilaments, and also the components of the cytoskeleton. So this is a contiguous system connecting the outside of the cell and the inside of the cell through the structural components of the membrane, as well as the structures found in the cell, and also the structures found in the extracellular side of the cell. So there are types of membrane proteins. These types recognize each cell. So you have cell-to-cell recognition. This is very important during development, which you'll be doing in year 4, developmental biology. That itself to a liver cell, for example, is because of the cell-to-cell recognition of these proteins. Like attracts like. Have the integrins, which we now know very much about. And then you also have intercellular junction proteins. Those of you who remember your cell biology, remember that between cells, there has to be communication from one cell to the other because cells are alike. And it's not just movement from one cell to the next. Specific junctions to control each other. And then you to move from one cell in a very controlled fashion. Then, of course, there's enzymes, which are okay. So you have enzymes which are also involved. Proteins, we know that enzymes by structure are proteins, and then we have signal transduction proteins because they receive signals from outside the cell, or even cells signals from inside and take them out. And then of course we have transport proteins, and these actually literally bind to a molecule and either passive you need to pay attention to all of this, and all of this has to happen using 1 to 4. So going forward, that's where we are going to pay a lot of attention to. And identify ourselves. What that's the reason I was telling you don't find a brain cell attached. So carbohydrate chains vary between species, individuals, and even between cell types in a given individual. So, like I said, liver cell carbohydrate chains are very different from kidney carbohydrate chains. Glycolipids also play a major role in cell recognition. So the glycolipids which are found in the plasma membrane are also very important in cell-to-cell recognition. Integrin, of course, are the integral protein to integrins on the cell, the seismic part of the membrane. And integrins play a very important role in strengthening the membrane. If you remember, the cytoskeleton is the top of a cell. It keeps the shape. That was cell biology 1114. So integrins bind to the cytoskeleton and maintain that shape so you don't have a blob of cell. And if you did practicals, you noticed that liver cells, kidney cells, brain cells are all different shapes. It is because of the integrins, which are very important in determining cell type. Then you have the intercellular junction proteins, and this helps cells which are alike to stick together to form tissues. So you don't have cells from one particular organ joining together with cells from another particular organ, a different organ, sorry. And then you have a total mess and confusion. And then I mentioned that many membrane proteins are enzymes. This is especially important on the membrane of organelles. And you know from your biochemistry that is this the lock and key hypothesis. This triangle here will fit in this part of this integral membrane protein, and this square here will fit in this square. So this can never bind here, and this can never bind here. So how does this happen? Intracellularly, the enzyme will bind here, activate this integral membrane protein, which will then make this able to accept this square, and then that will cause activation of other intracellular molecules inside the cell. And I think in your enzymology, you did something about enzymes which need to be activated in order for there to be a conformational change to activate that enzyme. So then you also have signal transduction or receptor proteins, which bind things like hormones and other substances on the outside of the cell. So these receptors will be found sitting outside the plasma membrane on the extracellular face. So the binding of the ligand or the signal, which is referred to as ligand, Ligand, will bind the receptor, and that binding will trigger a change inside the cell. And this change is called signal transduction. So take, for example, insulin will bind to insulin receptors which are found on the surface of cells. Once the insulin receptor has bound the insulin, you'll find the cell puts glucose transport proteins into the membrane. So these transport proteins for transporting glucose will not work until insulin binds insulin receptor. And you are aware that some diabetics have insulin receptors which do not function. So these are some of the importance of what we're talking about. So how does this receptor mechanism work? This is the ligand or the molecule or the hormone or whatever it is outside the cell. It will come, and you look at the shape here, and it will find its receptor. It will bind into the receptor, and look at the receptor. It's an integral membrane protein because it traverses the entire length of the lipid bilayer. Once it binds, it will activate, perhaps not always, but in many cases it will activate a molecule. That molecule will in turn activate another integral membrane protein, and that integral membrane protein, maybe it binds this in the inactivated stage. And once this signal comes through, it will release this molecule, and this molecule will go into the cell and start doing magic and start doing other things inside the cell. This molecule would never have been released from this or activated because of this if the receptor had not come. So you can have hormones coming all the way from the brain. You have the brain hormones which are released and go to and act on ovaries, and they travel in the bloodstream and go all the way down and activate receptors in I'm having a problem. This is just, there's a sound of crazy things going on here. Okay, let's try. Move to the next one. Okay. So compromisos. Transport proteins can, I hope you can hear me. So transport proteins are of different kinds. This is separate. We have the receptor protein. Now we have another category of proteins, and these are called transport proteins. There are two kinds, passive and active. The passive kind will allow water and soluble substances, which include small polar molecules and ions, to pass through the membrane without any energy cost. In other words, it will just kind of diffuse through. That's why it's called passive. Then you have active transport proteins, and the cell expends a lot of energy, and this energy is in the form of ATP, or adenosine triphosphate, and this will transport water, soluble substances against their concentration gradient. So that means outside the cell is low concentration, inside the cell is high concentration. And you know diffusion occurs in the opposite direction, from high to low. So that doesn't cost energy. But in order for you to move a substance from a low concentration to a high concentration, you're going against the passive transport rules of thermodynamics, which will cost you energy. And this energy in cells, as you very well know by now, is in the form of ATP. So how does this work? Look at the blue dots, that's high concentration, and inside it's low concentration. Look at the red dots, low concentration inside and high concentration outside. So you're literally moving these cells through a carrier protein or a channel protein, and you take it from the outside to the inside. It doesn't come in through here. It comes through what is known as the channel protein, and you can see this space in the middle is called the channel. In the carrier protein type, you have the molecule coming in, and it will bind in here, and when it binds in here, it will cause opening, and once it causes opening, the molecule will be moved to this side of the membrane. Of substances across the plasma membrane will happen through passive transport, which is simple diffusion, or facilitated diffusion, or simply by osmosis. Then you have also movement of substances across the plasma membrane by active transport, also bulk flow. Bulk flow happens when the membrane itself pinches and surrounds a whole bunch of molecules, and then it vaginates and delivers you. That you already know about movement of molecules across two phases with a membrane separator. Then you have facilitated diffusion, which I just showed you, where it happens through a channel protein or a transporter protein. And then of course you know osmosis, is there's active transport, which requires energy in the form of ATP. And then there's bulk flow. Bulk is of two kinds, bringing in, and exocytosis, out. And you know cytosis, cyto, referring the Latin for cell. So in endocytosis, you find the membrane invaginating and taking whatever material, forming surrounding that material and bringing it into the cell. In exocytosis, you have a vesicle which travels to the membrane, attaches to the membrane, and then the membrane opens up and releases the material, and that is called exocytosis. Typing. Can you hear me? Can you hear me? You can see my mic is flat. Okay, good. So we are now going to move to another presentation where we are going to look at some of these components in preparation for tomorrow's lecture. I think we have at least, and let us cross our fingers, that we shall be able to at least touch on some highlights so that we can have something to study tonight. We're not doing so badly, all things considered. Thank you for your patience. Okay. Yeah. So, first of all, when you talk about transportation, the transportation is known as transport. Polar molecules cannot diffuse across a lipid bilayer. Many such molecules, such as glucose, are able to cross the membrane, the cell membrane. These molecules then pass across the membrane through the action of specific transmembrane proteins. And these transmembrane proteins act as transporters. And remember, the transmembrane proteins that we've met so far, integral membrane proteins, the ones that traverse the entire membrane. So such transport proteins determine the selective permeability of the cell membrane, meaning the transmembrane proteins act for particular molecules. They don't act for all the molecules. They are membrane proteins for different substances that are needed to cross. And such transport proteins to selective permeability in membrane function. They contain multiple membrane spanning regions, and these membrane regions are the ones that form a passage through the lipid bilayer, and it is through this passage that allows that polar or charged molecules are able to cross the membrane through a protein pore. And when they do that, they do not interact with the hydrophobic fatty acid chains of the membrane phospholipids. So it is like a tunnel where they pass through. In fact, some of these transporters are called, as you come to find out in a few minutes. So transport across cell membranes of small dioxide from biochemistry, you knew that for metabolism inside the cell, they are soluble in the lipid bilayer and therefore can readily cross the cell membrane. Okay, small and molecules such as water, large uncharged polar molecules such as glucose cannot. So glucose cannot move from inside the cell to out or outside the cell to in. There has to be a stimulant and a transporter, to move glucose. And we'll look into this in a little bit more detail. So charged molecules such as ions are unable to diffuse through the phospholipid bilayer regardless of the size. Even protons, hydrogen ions, can layer by free diffusion. What moves in and what moves in is tightly controlled through the phospholipid bilayer. I have a question here from Mwekesa Wanyama. Sorry, I've just seen it. What is the process through which Bang content flows into the, and what does it do for one to become high? My friend, the Bang content, those molecules have to cross the membrane bilayer. And it is for these, I'm talking about small uncharged molecules all the way to the large molecule. So you know Bang is one of those amines, and we are going to talk about that in a little bit more detail later on when we come to those brain molecules. However, we'll talk about the large molecules here. You'll be able to understand how the Bangi crosses from the blood to the membrane and into the brain cells and drives you crazy and you become bonkers. So, Wanyama, Wekesa, we really don't want to be doing that. So small uncharged molecules such as dioxide, carbon dioxide is a byproduct of cellular metabolism. Cellular metabolism requires oxygen and water, and petrol and ethanol can all cross the membrane. So this is are able to freely diffuse through the phospholipid bilayer. Okay. Large polar molecules, Mr. Wekesa, such as glucose, protons, cannot. The small and very easy. Today. There we go. So the permeability of phospholipid bilayers is past things like benzene, hydrobiolics through, but it's very dangerous. Molecules such as water, ethanol pass through. That's why people who think those illicit using wherever they are, the ethanol just passes right through. Bad idea. Large molecules such as glucose, amino acids, and charged molecules can pass through the lipid bilayer without carrier proteins or specific controls. I draw your attention to these molecules. They are small, but they are charged. Protons, chloride, sodium, calcium. Single charge, double charge, negative charge, positive charge. Doesn't make any difference. They're unable to cross the cell membrane just like that. So what are these transporters that shuttle these across? Transporters are distinguished from channels because they catalyze. And by catalyze, we mean mediate the movement of ions and molecules. And
hata Kristo anapiga bao. Si siri. Sio kama na Sambina
assembly.
Cristiano anapiga bao. Lazima. Ni leo ama? Si ni leo.
By kinetic parameters, which is applied to study enzyme kinetics. Transporters also exhibit specificity, which is another speciality which is found in enzymes, molecule being transported, as well as to define kinetics in the transport process. So if I was to ask you to explain to me the similarities between transporters and enzymes, you talk about the defined kinetics as well as the specificity. Without specific examples. So transporters also both and non-competitive inhibitors. Transporters are known as carriers. Their specific names: permeases, which enable things to permeate; translocators, which translocate the other; translocases, which is translocators but which are enzymes. Ases, we know that is an enzyme. And then porters. Literally, a porter carries your luggage from point A to B. So the porters on the plasma membrane move material from point A to B, or B to A, or B to C. So mediated transporters are classified based on the stoichiometry, and you know stoichiometry is shape and structure of the transport process. So there are three kinds of mediated transporters known as uniporters, symporters, and antiporters. Uniporters, see the name uni. Symporters will simultaneously transport two different molecules at the same time. Then molecules in opposite direction, from outside and from inside, different molecules, different directions. So what is a uniport? Integral-membrane protein which will transport one molecule in one direction at a time. Symporters, integral-membrane protein transports two different molecules in the same direction. Antiporters, two different molecules transported in different directions. Are we clear on that? Ladies and gentlemen, are we clear on that? This is very important. I would like you to copy it and have it at your fingertips at all times going forward. Please copy that and have it at your fingertips at all times going forward, because God forbid we come back to having to explain what uniport, symport, and antiport is. Good. I can see most of you have got it. Chip, 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 chip. Geez. I've been talking too much today. My tongue is gone off. So copy that. I'll give you 3 minutes.
Mimi ndio najua. Obwoti kuyuni komatindoi class amut mi akot netesetai nguni mpaka 6. Mi netesetai nguni mpaka 6. Nonon degree ka record. Kor baadaye kityo asub kit nekamwa. Kami Kikuyu. Komakwerichi tugen. Quite 206.
You are now unmuted.
Okay, we proceed.
Konoitu iwal again.
So, how does this happen? You have a channel protein. A channel protein will move a molecule through the channel in the middle. You see, the channel protein is an integral membrane protein, and in this case, you can see it is several proteins, this one, and that one, all of them forming a circle. And inside that circle, you'll find a pore that allows molecules to move right through. Then a carrier protein, also shaped like the carrier protein, integral membrane protein, but in this case, the external molecule will come, bind in there, and then this protein and formational change, you remember that from biochemistry, and it will open up. Once it has bound here, it will cause opening of this part, and molecule will move to the other side. Then, suppose you have low concentration of protons outside. This will require ATP, and ATP will cause the protons to move in because the inside just a minute, let me try and there we go. There we go. So you have high concentration of protons inside and low outside. And you know, in diffusion, you cannot get movement from low to high. Osmosis usually get from high to low. So in order to move these protons from an area of low proton concentration, you require ATP. And remember molecular biology, adenosine triphosphate. And here these three dots are the three phosphates. And once the phosphate binds on a very specific site on the integral membrane protein, you get ADP, adenosine diphosphate, and you can see two dots here. Those dots are the two phosphates, hence adenosine diphosphate, because one phosphate has bound on the integral membrane protein. So the proton binds in here just like the carrier protein. The difference being, in this case, you need energy because you're moving from a low to high concentration gradient. And then the proton will be released because there's a conformational change. And then the phosphate, which was bound here, is released from the binding site on the integral membrane protein. So in this one, there's three different levels. There's the movement from high to low. There's the requirement of energy through ATP. And then, of course, the phosphate molecule is released, and then the conformation of the carrier protein changes back to the original conformation, awaiting the next molecule that needs to be moved from one area to the next. Now, I'm not going to give you time to copy this because this is in your reading assignment. Those chapters and pages, I've given you specific pages. There are not too many. Some are 5, some are 10. So you'll be able to get these diagrams there. And I would like to do that so that I can force you to actually go and read. So this is just to explain what is happening. Then there is passive transport. Passive transport, you have the integral membrane protein in one state, this state, and then this is the closed state where it closes on the outside. This is the lipid bilayer. Now we are not drawing the lipid bilayer how you've become accustomed, but now henceforth we know this is the lipid bilayer. So a solute will come and bind a specific area in the carrier protein, which is supposed to mediate the transport of this particular molecule, solute A. And once it binds in there, it closes, and this is the solute binding site. It will close, and once it closes, it will release the solute. And in this scenario, you have movement of molecules from high concentration gradient to low concentration gradient. You will note in this movement transport, there is no metabolic energy, no ATP is needed because the solute is moving down its concentration gradient from high to low. Okay? Any questions? Mute or type? Have we understood that? I need to know before I move on. Having taught this for many years, I understand that sometimes. Okay, good. Sam has understood. Good job. Abby has understood. Good job. Any more? No? All right. I'll take you to and move with you. So, in passive transport, there's also a mechanism known as facilitated diffusion. So, outside and inside the cell, plasma membrane, the lipid bilayer. So here you have positively charged molecules which are moving inside through an electrochemical gradient which has no membrane. What is the membrane potential here? The outer part of the plasma membrane is positively charged and the inner part is negatively charged. So the electrochemical gradient has a membrane potential, positively charged out here, negatively charged, and you get the ions moving in to the cell because it is negative inside. Looking at the other alternative, if the membrane is negatively charged outside and you have positive ions that want to move in, and positively charged membrane inside, the electrochemical gradient with a membrane potential which is positive inside. So facilitated diffusion happens with these scenarios. Electrochemical gradient of the membrane. On the outside positive, on the inside negative, or alternatively on the outside negative and positive on the inside. And these scenarios happen in different tissues at different times. For example, you can find that the nerve cell is negatively charged inside because of the pre-existing conditions, or you can find muscle cells where you have negative charge outside the muscle cell and negatively charged inside the muscle cell. And if you've noted, I've given you an assignment to look at some of these things in the kidney, and I have said specifically epithelial cells of the proximal convoluted tubule of the kidney. And there is a very clear reason why I picked that. So that is my usual trick questions to make you think. So I'm just going to give you that hint. When you're doing your assignment, think about this carefully. So Crohn's affect both uncharged and charged solutes, and electrical gradient affects only charged solutes, and this is what forms the electrochemical gradient. I repeat, concentration gradient affects both uncharged and charged. Remember, we talked about moving from high to low, from low to high for the protons. So whether they are charged or not, they will be affected by concentration gradient. But electrical gradient will affect only charged solids, and then it is named electrochemical because of the electrons, whether they are charged or uncharged. So how do you define active and passive transport? Passive transport, there is no metabolic energy needed because the is moving down its concentration gradient. In the case of uncharged solute, the concentration of the solute on each side of the membrane will dictate the direction of passive transport, positive or negative. In active transport, metabolic energy in the form of ATP is used to transport a solute from the side of low concentration to the side of high concentration. So this is just a summary of what we've talked about in the last 5 slides. Now, what's the difference between passive transport and active transport? And I put this here because we learned it in this book. Again, the same book which we used for molecular biology. The lipid bilayer, simple diffusion, movement of molecule, this little black dot across the membrane. Those will open and allow the molecule to pass through, and that is referred as channel mediated, and it is the first type, of passive transportation. The second one, which we talked about, was the carrier. So this molecule will bind to the site, which is specific for that molecule. You'll have a conformational change, opening of the integral membrane protein or the carrier protein, and the molecule will pass through. Carrier proteins are also used in active transport, but in this case they require G, and that will mean movement of this molecule across the membrane. And if you look at this here, this is a depiction of the concentration gradient. This molecule, which is being moved through this carrier protein, is moving from a low to a high. So this defeats logic when you think in osmosis, because in osmosis you have movement from high to low. But in this case, you will require energy to move this molecule against its concentration gradient in this direction. Are we clear on this? On the 3 types: diffusion, channel mediated, carrier mediated, and also distinction between passive and active transport. Those are 3 concepts which we've talked about, all put together in one figure. Can I have feedback, please? Yes or no? Good. Nyamwaya, good. Yes. Cheboyo, Kyoko, Abi Osman, very good. So we move on. Thank you. So free diffusion and facilitated diffusion are two types of diffusion. So far to this point of your education, you've only known about one type of diffusion, didn't make any difference, but there's the free variety and the facilitated variety. So in free diffusion, you have two types. Non-channel mediated, the one which I showed you, lipids and gases just passing through the membrane. And then you have channel mediated, where you have ions and charged molecules passing through that tube, the channel, the tube which is the membrane proteins. Then in facilitated diffusion, it is carrier mediated. All the amino acids shifted from one side of the plasma membrane to the other. So that is the distinction. Free diffusion can either happen across the membrane with no channel involvement or with a channel mediating the movement. Facilitated diffusion must be facilitated by a carrier, and I have shown you how the carrier is involved in the movement. So this channel, and this is a carrier, and example of facilitated diffusion for this molecule here in yellow, moving it from inside, because this is the cytosolic phase and this is the extracellular space. So this channel will allow the movement of this, and it is facilitated by this pore. Okay? The carrier, on the other hand, will bind the yellow molecule here. You can see it's closed on the outside, and then a conformational change will result, and this will release the molecule out. So this is how facilitated diffusion. Yeah, each of this molecule has released this original conformation until another molecule comes and binds. Here on this, can I response? And then are we all on board? Can I have feedback, please? Yes, no, maybe? Okay. Can you hear me? Amatete? Good. Thank you. Oma? Good. All right. So, two classes of proteins that mediate facilitated diffusion are generally distinguished as carrier proteins and channel proteins. So you have this molecule which is coming in here. It's trapped in there because of the conformational change of this integral membrane carrier. Then the conformational change releases this. So this is referred to as carrier-mediated solute transport. Very similar to the last slide, just a bit of artwork. So channel proteins form open pores through which the membrane, allowing free diffusion of any molecule of the appropriate size and charge. On the other hand, carrier proteins are responsible for the facilitated diffusion of sugars, amino acids, nucleosides across the plasma membranes of most cells. You know what nucleosides are. So, in contrast to channel proteins, carrier proteins selectively and transport specific small molecules such as glucose. Rather than forming open channels, carrier proteins act like enzymes because they have to bind a specific place on the carrier proteins. Remember that lock and key hypothesis of enzymes, and then remember the kinetics of enzymes. So this happens to facilitate the passage of specific molecules across membranes. In particular, carrier proteins are known to bind specific molecules, then undergo conformational change. This change will open the channels through which the molecule to be transported can then pass across the membrane and then be released on the other side. So I would like us to leave it here. We're on slide number 21, up here tomorrow, and then I will give you time to answer or ask at this time questions. Let me ask the first question. How is this compared to molecular biology? Are you getting it? Or am I just talking to myself? You can unmute or you can type. Hello? Are we there? Is this easier than molecular biology? No? It's easier. Okay. Yeah, that tells me that you're understanding it easier, better. That's good. That's good. It's better off, okay? Sylvia?